Okay, hello everyone. I'm Dr. Christopher Jones, and this is the second lecture of MD4008. The topic of the lecture is bacteria. So for this mini series with which we're opening MD4008, we're working through the groups of microorganisms one by one, and we're essentially gaining an understanding of what these microorganisms are, getting all the key bits of information that we need to get about them before we progress through the module and look at these microorganisms in the context of human health and industry and the immune system and all these complex things. So today the focus is bacteria, so hopefully you'll be learning exactly what bacteria are. So we can start off by defining bacteria, and this is some information that we covered last week. It's just the idea that bacteria are prokaryotic cells as opposed to eukaryotic cells. And again, it was covered in the slides last week. If you're not clear on the difference between prokaryotic cells and eukaryotic cells, you just need to sit down, study the information and make sure it's really clear to you what the difference is. The information, again, as I said, is in the slides last week. I don't want to go over it again. It's not a good use of our time, really. Yep, so just go back to those slides if you're struggling. When we're defining bacteria, we need to talk about their gram status. So something I referred to last week as well is the idea of gram-negative bacteria and gram-positive bacteria. And this classification is dependent upon the cell wall composition of the bacteria, essentially. We're going to go into a lot of detail about this today, so I'll leave it at that for now. But this gram status is really, really important, and we will gain a good understanding of it today. We'll also gain an understanding of why it's important. It's not enough to understand what we mean by gram positive and gram negative. We also need to understand why that matters, so why we even bother talking about gram positive and gram negative, why we use the definition. This is also something we briefly went over last week. So we discussed the different morphologies of bacteria. We said essentially bacteria can look circular, spherical, and that's the cocci morphology, or they can look like bacilli, which is like a capsule shaped, like a tablet. Then we have the spirochetes. So these are bacteria that look almost like a squiggle. Again, that's all the information we really need there. This is a diagram I showed you last week when I was talking about the relationships between the different forms of life. And this is a really, really important diagram. The actual diagram I showed you last week was different, but the information was the same. The information detailing the relationship between the three kingdoms, archaea, bacteria, and eukarya. Sorry, the three domains, not kingdoms. It's super important we understand this. We need to understand that all eukaryotic organisms belong to this eukarya domain. All bacteria belong to the bacteria domain, and archaea are in their domain on their own, tucked away, that we don't really worry about. The other key bits of information we need to understand relating to this are the fact that archaea and bacteria are both prokaryotic domains. So members of the archaea domain and members of the bacteria domain are all prokaryotic. What that means is they have cell compositions which are prokaryotic, which again we just talked about, and it also means they're unicellular. So organisms that belong to these two domains are composed of a single cell, and that single cell is prokaryotic. The eukaryotic domain, however, is a lot more complicated. We have a lot of multicellular organisms in this domain. We also have single-celled organisms, so we get single-celled organisms and multi-celled organisms that are eukaryotic. However, the key point is all these cells, all these organisms are eukaryotic, so they are composed of one or more eukaryotic cells. And again, as we talked about last week, eukaryotic cells are fundamentally different to prokaryotic cells, and that's something we need to understand. Okay, again here we're grouping archaea and bacteria together because even though they're two separate domains of life, they are both prokaryotic. They are both composed of single-celled organisms. Eukarya, a third domain of life, very different from bacteria or archaea because members of the eukarya domain are eukaryotic, meaning they're composed of one or more eukaryotic cells. And here we have those distinctions just typed up nice and clearly. So again, we have eukarya on the end, and we have the definitions of eukaryotic cells listed there. This is the same information in graphical form. So again, if you're struggling with this information, study this diagram. I'd encourage you to access the textbook, MIMS Medical Microbiology, from which I got this diagram. It has some really nice accompanying explanations, very clear. Some of the information in the textbook is beyond level 4. It's really a level 6 textbook, but nonetheless, it's still useful and I'm sure you'll get some value out of it. So a lot of what I've talked so far about today has been a bit of a recap of last week. There's been bits of extra information, but it has largely been a recap. So what I want to do moving into the lecture is just go into some more specifics, provide some new information, provide some real depth of understanding of bacteria. Everything we've talked about so far, it's super important and we need to understand it, but it's quite shallow, it's quite surface level. It's stuff that should be quite simple for you. Now we're going to go into a little bit more detail and actually understand just in a bit more depth what bacteria are, look at concepts a bit more thoroughly. And first, we're going to do that with this idea of gram-positive and gram-negative bacteria. I kind of alluded to the importance of this classification of gram-negative and gram-positive bacteria. I said it exists. I said we need to understand it. What I want to do now is actually explain the distinction from a biological perspective. So what is the difference between a gram-positive cell and a gram-negative cell? The answer lies in the cell wall of the bacteria. So gram-positive bacteria essentially have this thick peptidoglycan layer of their cell wall. So their cell wall is just a thick chunk of peptidoglycan all around the outside of the bacteria. There's consequences to having this thick peptidoglycan wall that we'll talk about later. For now, we just need to know that it exists and it is gram-positive bacteria that have it. Gram-negative bacteria, therefore, don't have a peptidoglycan cell wall. They lack one. What they have instead is a thin layer of peptidoglycan. As you can see in the diagram that's up now, you can see the gram-negative bacteria just have this thin peptidoglycan layer. And outside this layer, they have a layer of lipopolysaccharide, but it's a bit more complicated than that. So this outer layer is largely composed of lipopolysaccharide, LPS, but there's other bits and bobs in it as well. So that's the key difference. It really is as simple as that. Some students will get confused between the two. Try not to. Again, this is such a fundamental concept, you really shouldn't be getting confused. Gram-positive bacteria have a peptidoglycan cell wall. The best way you can remember it is they're positive because they have something the negative cells don't have. And what they have is a thick peptidoglycan cell wall. Gram-negative cells are negative because they don't have something that gram-positive cells have. In place of that, they have just a thin peptidoglycan layer. They do, however, have LPS on the outside of that. So the outermost layer of the gram-negative bacterial cell wall is lipopolysaccharide mixed with a bunch of other stuff. So hopefully that makes sense. This is another diagram showing the same thing. Again, some students connect better with different diagrams for whatever reason. So we're looking at exactly the same thing, just a slightly different representation. We have gram-positive bacteria on the left. Again, you can see they have this thick peptidoglycan wall. And then we have gram-negative bacteria on the right. And again, you'll see this very thin peptidoglycan layer, and outside this peptidoglycan layer, we have this lipopolysaccharide layer. So we have this LPS kind of outer membrane on the outside. Okay, so next we need to talk about gram staining. So I've told you we have gram-negative bacteria and we have gram-positive bacteria, and later on we'll talk about why that matters. But for now, we need to talk about how we can tell the difference between the two. It's one thing to say that gram-negative bacteria exist and gram-positive bacteria exist, but how do we tell the difference between the two? If we can't tell the difference between the two, there's not even much point in making the distinction, surely. Luckily, we have a way to tell the difference between gram-positive and gram-negative bacteria, and it's a very good way. It's a lab-based method that's been used for a long, long time, that's still used in the labs. It's called gram staining. You've probably heard of it. You may have done it before. What I'm going to do right now is just talk you through the basic principles. So talk you through the fundamental steps that compose a gram stain and talk about why we do each of those steps, what purpose each step serves, and hopefully with your understanding of gram-negative and gram-positive bacteria, so your understanding of the differences in cell wall composition between the groups, the gram stain will make sense, and it will make sense why we do each individual step. So we'll go through the steps of the gram stain one by one. First off, there's a practical step. We're taking the bacteria we've grown. So we're imagining we're in a laboratory environment and we're smearing the bacteria onto a glass slide and then fixing them in place just using heat. So we call it a heat fixed bacterial sample. Once that's done, we add crystal violet. Now, crystal violet is our first reagent, and what crystal violet is going to do is stain the bacteria purple. So we call it our primary stain. So we're using crystal violet as our primary stain. And again, what we're staining is the bacteria, and we're staining them purple. Once we've stained them with crystal violet, we're then adding iodine. Iodine is acting as a mordant, so it's essentially fixing the stain in place. It's fixing the dye, and in doing this, it's forming crystal violet iodine complexes. These complexes are essentially what are formed, and they're what fixes the dye in place. The formation of these complexes is the fixation process. Okay, then we need to add alcohol acetone, which is a decolorizer, and this is where we get a differential effect between gram-positive cells and gram-negative cells, because alcohol acetone will not decolorize the gram-positive cells. The reason is the peptidoglycan layer just prevents the process from occurring. Because the gram-positive cells have this thick peptidoglycan layer in the cell wall, the alcohol acetone just isn't able to decolorize the cells, and the cells retain the purple color. In the gram-negative bacteria, however, and again we're assuming we're looking at a sample with a mix of gram-negative and gram-positive bacteria, so in gram-negative bacteria, the alcohol acetone decolorizes completely. So it gets rid of the primary stain, gets rid of the crystal violet, and you end up with uncolored bacterial cells. We then add a counter stain called safranin. Safranin is going to stain the gram-negative cells pink, and it's going to do nothing to the gram-positive cells. The reason it's going to do nothing to the gram-positive cells is because the gram-positive cells are already purple and they have color, but the gram-negative cells, however, they're colorless. So when you stain them with safranin, you can very clearly see they have this pink color, which is obtained from the safranin. If we look at a diagram in which we're doing the gram stain, so we have the individual steps represented on a diagram, we can see the process we've just described play out. In this example, we have a mixed population of bacteria, so we have gram-positive bacteria mixed with gram-negative bacteria. You could see this in a practical environment. Often in the lab, you're isolating a single bacteria from a specimen, and then you're culturing that bacteria. So often when you're doing a gram stain, it will be on a single species of bacteria. So all bacteria in the sample will react the same way and end up the same color. Not always, however, and in this example, in this diagram, we're looking at two mixed populations of cells. We're looking at we're looking at a mixed population of two types of cell. We're looking at cocci, so some bacteria that are circular, and we're looking at bacilli as well, so some bacteria that are elongated. So the first step is to apply the primary dye or the primary stain. This is crystal violet, and crystal violet will stain all bacteria purple. So regardless of whether the bacteria are gram positive or gram negative, they're going to stain purple at this stage. The second stage is to add iodine, and iodine is acting as the mordant. So again, what iodine is doing is it's fixing the primary stain in place. It's mediating the formation of crystal violet iodine complexes, which is the fixation process. After this step, alcohol wash is added. So we use alcohol acetone. And what alcohol acetone is going to do is it's going to decolorize gram negative cells. It removes the purple color from gram negative cells by essentially washing out crystal violet. It doesn't do anything to gram positive cells, so it doesn't successfully decolorize gram positive cells. The reason for this is the presence of the thick peptidoglycan cell wall. So we've said gram-positive cells have this thick peptidoglycan cell wall. Because of this thick cell wall, they retain the purple color. They retain the crystal violet iodine complexes. The alcohol acetone doesn't decolorize them successfully. Gram-negative bacteria, on the other hand, are successfully decolorized because they don't have that thick peptidoglycan cell wall. They have just a thin peptidoglycan layer surrounded by an LPS envelope, and the alcohol acetone disrupts the LPS envelope anyway. So there's really no hope of these bacteria retaining the CVI complexes. So they very quickly lose their color and become colorless. In the next step, we add saffron in, which is the counter stain. What we see when we add saffron in is the gram-negative bacteria are successfully stained pink because at this stage they're colorless because they've been decolorized by alcohol acetone. The gram-positive bacteria, however, aren't stained pink because they're already stained purple, so they've retained the CVI complexes, so they retain their dark purple color, whereas the gram-negative cells turn nice light pink. This is what that looks like. This image on the right is just a bit less cartoony than the one we looked in the last slide. You can clearly see the circular cocci, and you can also clearly see the elongated bacilli. Here they're labeled as rods. Rods, just kind of an informal term for bacilli, because I guess they are kind of shaped like rods because they're elongated. In this example, you can see the rods are gram-negative and the cocci are gram-positive from the color pretty clearly. This is it kind of looks like a dark blue, but it is a purple for the cocci, so they're stained with the crystal violet purple, whereas the gram-negative rods, we can see they haven't retained any purple color. They're quite clearly pink, and this pink is coming from the saffronin counter stain. This is what you see under the microscope when you're finished doing a gram stain and you're looking at the results. It's pretty clear, and it's often as clear as this. Sometimes it's not as clear as this. It depends how polished your technique is. As with a lot of these scientific techniques, even though they are structured, even though it is systematic, there is also almost an art to getting it right, and it's just experience. It's just how well you do the individual steps. Are you making mistakes? Are you doing things in a timely manner, or are you taking just a bit too long? Are you sticking to the kind of time limit suggested for each immersion? So are you leaving the crystal violet on for the right amount of time? Are you leaving the iodine on for the right amount of time, or too long, or not enough? It's about fine-tuning these little things. When you start to do lab techniques like this, you'll see that's the case. You'll see that's the case. But this is a very, very clear example of what a gram stain should look like, or what the results of a gram stain should look like. Next, we want to answer the question of why all this matters. So why do we care if a bacteria is gram positive or gram negative? Why are we putting all this kind of emphasis on gram staining? Why do we need to determine this information? The answer is because it has implications that really, really do matter. So by determining if a bacteria is gram positive or gram negative, we gain information that is useful to us. An example of this is susceptibility to antibiotics. So different bacteria have different degrees of susceptibility to different antibiotics. Gram-negative bacteria in particular are resistant to some antibiotics, and it's useful for us to know about that. Again, I understand we're not clinicians and we're not determining how to treat patients, but we are potentially going to be working in labs. We are potentially going to be dealing with lab specimens and patient samples, and we're going to be trying to determine, what bacteria are in those samples. And one of the most useful bits of information you can obtain is whether the bacteria present is gram positive or gram negative. Firstly, because it helps to inform decisions on how to treat infections caused by that bacteria. Secondly, and this is related, this is also to do with diagnosis, and this is to do with determining the exact specific causative agent of an infection. So we have the gram positive status of cell or the gram negative status of the cell confirmed. We've done the gram stain. We know if the bacteria is gram positive or gram negative. That gives us an indication of how we could potentially treat the infection. That's not the end of the story, however. There are then a multitude of other tests we could do that help us determine exactly which bacteria we're looking at. So we're going in way more depth than just the gram stain. We're understanding that gram stain is just the start. And once we've done the gram stain, we can then do a whole number of additional tests that again let us effectively diagnose and determine what the exact causative agent of an infection is. Obviously, this is super important because it gives us even more information that will inform therapeutic options. So what antibiotics we could potentially use, as well as other information. It can give us ideas about what symptoms we can expect, things like that. Again, we're not clinicians, but this is useful information that could potentially inform a clinician. So while we're just focusing on the element of diagnostics that is to do with determining what the bacteria is, so what species we're looking at, it's useful for us to be aware of these wider concepts. Here we see a fairly clear demonstration of the importance of gram staining and determining the gram status of bacteria in the process of diagnosis. What we're looking at is a diagnostic flowchart, essentially. Well, really it's two diagnostic flowcharts, and the determination of which flowchart to use is entirely dependent upon whether the bacteria being studied is gram positive or gram negative. So clearly it's an important factor, it's an important characteristic. When you're looking at these diagnostic flowcharts, you'll see almost always the first step is gram positive or gram negative, because we consider these two groups of bacteria quite separately. So yeah, when doing these diagnostic flowcharts, when determining a diagnosis, when trying to specifically decide what bacteria you're looking at, almost always the most useful thing you can do is determine whether it's gram positive or gram negative first. You'll see as we progress down the flowcharts and get towards the individual species, there's other tests being done. It doesn't really say what tests are being done, but it says the outcomes of the tests, so whether or not bacteria is aerobic or anaerobic, whether it's coagulase positive or coagulase negative. Don't worry about these for now. I just want us to understand the principles for now. What we need to understand is that each of these arms of the flowchart is a test that's being done. We're determining if the bacteria behaves this way or this way under these conditions. We're determining if it looks like this or like this under a microscope. So we're kind of determining the classification of the bacteria. We're getting more and more information about the bacteria that allows us to ultimately arrive at a specific identification, so to specifically determine what bacteria we're working with. One thing I do want you to notice is the first branches of the flowchart. So each flowchart we have cocci and rods for the gram positive, and we have the same for gram negative. It's backwards, but it doesn't make any difference. It just says rods and cocci. These refer to the bacterial morphologies we were talking about earlier. So there's a reason why we looked at that information right at the start, because just like the gram status, just like whether a bacteria is gram positive or gram negative, the morphology of the bacteria is important because it's helping us to determine what bacteria we're looking at in a diagnostic situation. So if we have bacterial samples coming into the lab, first we're going to determine if the bacteria is gram positive or gram negative. Then we're going to determine if it's cocci or if it's the term here is rods, but bacilli is the technical term. So we're going to determine whether it's cocci or bacilli, or in some cases spirochetes. Interestingly, if we look at the gram negative arm, and this is almost an aside, but you can see there's only one cocci, so that is gram negative. So if we get a sample from, let's say we get from a urine, so it's a urine sample, we get a urine sample, we determine the bacteria present are gram negative, and then we look at them under the microscope and we see their cocci. There's there's two possibilities of what it could be. Usually it's going to be Neisseria. But yeah, the point there I'm making is just from those two little bits of information, we're already very, very close to a diagnosis. So that's how useful this information is, it's how powerful it is. I think we've covered gram negative and gram positive differences and why they matter and all that kind of stuff. So we'll move on from gram staining and gram positive and gram negative classifications and look at some of the other features that we find in bacteria. Firstly, I want to talk about the capsule. So the capsule is something that surrounds the bacterial cell wall. It's just a mesh of polysaccharide and protein. It's sometimes called a slime layer. It depends upon the kind of constitution of it, how well defined it is. Sometimes you'll get a very, very well defined capsule. Sometimes you'll get a less well-defined capsule, so it's just a bit more slimy, a bit looser. That's what we tend to refer to as a slime layer. The capsule is interesting in that it tends to be used in attachment. So when we're looking at infectious disease specifically, so when we're looking at how bacteria cause infectious disease, you'll notice how important the capsule is, specifically in the process of attachment, and you'll also realize how important the process of attachment is to infection overall. You'll also realize that the capsule is important purely because it's on the surface of bacteria. So just by virtue of being on the outside of the bacteria, it's important because it's what our immune system is coming into contact with first, for example, and there's implications of this. So again, for now, let's just remember that it exists. Let's just remember it as a bit of a mishmash of polysaccharide and protein. And yeah, we don't need to know much more than that for now. The fimbriae, flagella and pillae, however, we do need to know a little bit more about. Really, we just need to know the distinctions between the two, the three, and understand, you'll see why I said two in a second, but understand the differences between them, understand what they're for, and by what they're for, I mean what their purpose is, why they've evolved. Nothing too complicated, we just need to understand it. Okay, first off, we have the fimbriae. These are primarily involved in attachment. They're present on the surface of bacteria. They're protrusions on the surface of the bacteria that kind of stretch out into the environment, and that's the role in attachment, because they're reaching out almost into the environment. They're the first point of contact between the bacteria and the surfaces the bacteria are coming into contact with. Again, like the capsule, they have a role in pathogenesis because they have a role in attachment. Pillae, these are less numerous and these are longer, so you can think of fimbriae as being almost all over this. I wouldn't say all over it, but there's lots of them on the surface. Pillae, there's less of them and they reach out further. Pillae can function in attachment as well. It's not their primary function, but they can function in attachment. More importantly, they function in genetic transfer. The reason why I said 2 instead of 3 earlier is because fimbriae are technically pillae. Pillae are just, it's kind of a general term for protrusions that can interact with the environment in some way. And they're even composed of the same things. They're composed of what we call pillins. Pillins, you can think of a pillin as a subunit, and the subunit is repeated over and over and over and over again to form a pillae, and some of those pillae are fimbriae. Okay, again, the key thing about pillae isn't their role in attachment, even though there are pillae called type 4 pillae which do act in attachment. We're more concerned with the sex pillae. They're called sex pillae specifically when we're talking about E. coli. More generally, they're just called pillae. Sometimes they'll be referred to as conjugated pillae. You'll understand in a few slides. For now, just understand that pillae are involved in genetic transfer and a very specific mechanism of genetic transfer. Flagella are distinct from pillae in that their primary function is movement. Flagella, here we're talking about bacteria, but you can also have flagella on eukaryotic cells, on the flagellates, and the role is the same. You can almost think of them as like a tail, and it's not a bad analogy. If you look at diagrams, you'll see it tends to look like a tail. If you look at EM images as well, you can see it kind of looks like a tail. And again, the idea is propulsion. The flagella just allows movement or allows taxis in a particular direction. Here we have some information on genome organization, and we've touched on the fact that bacteria don't have a membrane-bound nucleus, which eukaryotic cells do have. Bacteria instead have a nucleoid, which is just a bit of a jumbled, I shouldn't really say mess, but a jumbled region of DNA. There's more to say, however. They do have this nucleoid, but they also have accessory genetic information, namely in the form of plasmids. So bacteria, like all prokaryotes, have the potential to carry plasmids, which are replicating circles of DNA. So these circles aren't part of the main nucleoid, so the main part of the genome that's included in the nucleoid. However, they are transcribable and translatable. They are functioning. They're just kind of floating around separately, and again, they're circular, so they're enclosed. These are independently replicating. So these are capable of replicating independently of the nucleic acid, which is contained loosely within the nucleoid. So hopefully those differences in structure across bacteria and other cells are clear, and we have a clear idea of what the structure of a bacterial cell is like. Now I want to talk about bacterial reproduction. Super important topic. Bacterial cells don't reproduce in the same way as eukaryotic cells, and we need to understand that, and we need to understand it in detail. We need to understand exactly how bacterial cells replicate, how bacteria reproduce, and we need to understand the implications of these reproduction mechanisms. This information is something you may have heard before. It's what we tend to think of when we imagine bacterial replication. It's the idea of binary fission, essentially bacteria splitting in two, bacteria doubling. You remember the graph I showed in the last lecture where we saw exponential bacterial growth. So we saw a graph of bacteria growth, so the axis measured time versus bacterial numbers, and we saw that over time the number of bacteria doubled and doubled and doubled and doubled. It hit a plateau, the stationary phase, but for a period of time it just doubled and doubled and doubled. This is exponential growth, and this is possible because bacteria are capable of reproducing via binary fission. They are capable of copying their own genome, so producing a copy of their genome, and then essentially splitting in two. By this mechanism, it's pretty clear to see how bacterial numbers can double. And again, that's what exponential growth is, doubling over a period of time. And this period of time can be quite short, sometimes. So between 1 and 3 hours is a pretty good estimate, but it can even happen quicker than this. Worrying if you're trying to control bacteria growth. Again, do bear in mind that there is that upper limit. There's the stationary phase that is eventually reached. So this exponential growth isn't going on indefinitely. It has limitations. It won't continue forever. I've put some of these limitations on the slide, including environmental nutrients, metabolic byproducts, competition, competition by other bacteria, but also competition by the other members of the same species that are being produced by binary fission. If you have this doubling of numbers every so ever many hours, each of those bacteria, they're in the same location, they're going to be competing for the same resources. That competition is going to limit reproduction because, again, there's only so many nutrients to go around. Okay, hope that all makes sense. The diagram on the right should explain it as well. It's literally just they split into, they copy their genome, so you have an exact genome copy, and then when they split in two, binary fission, split by two, binary fission. Each copy of the genome will go to a separate daughter cell, so all the information is retained. There's no loss. Binary fission is only part of the story. However, it's not the only method of reproduction available to bacteria. There's inherent issues with binary fission, which mean that bacteria can't rely upon it solely. The main issue is that it doesn't allow for genetic recombination. So when we're thinking about binary fission, again, it's a single bacteria splitting in two. There's no new genetic information being introduced there. Each of the progeny are identical to the cell that produced them. That's not a great evolutionary strategy because it doesn't allow for adaptation to the environment. it's limiting. You end up with a huge pool of bacteria that are, with the exception of random mutations, identical. So by and large, their genomes are identical. Again, that's a limiting strategy. You can imagine it's quite easy for an entire population of these cells to be wiped out because they have the same susceptibilities. They ultimately have the same genome, again, with the exception of potential random mutations. It therefore benefits bacteria to have a means to induce genetic recombination, so to produce progeny that are not identical, or to be amenable to mechanisms that change the genetic material of cells to make sure they, again, to introduce new genetic material to allow for genetic recombination. And this is really important because, again, binary fission, it's not sexual reproduction. When we think about sexual reproduction, we're thinking about genomes from two members of the species joining together, recombining to produce a progeny that is genetically unique, that is genetically distinct from the members of the species that produced it. Again, this is really, really powerful, evolutionary speaking, because it allows for adaptation. You end up with a population that is, again, genetically varied, not identical to each other, and there's real power in this, speaking from an evolutionary perspective. So it's not surprising that bacteria, despite relying heavily upon bacterial fission, have evolved alternative means to produce genetic recombination into their progeny. 3 of those means are outlined in this slide. So we have transformation, transduction, and conjugation. We're going to talk through each of these, explain exactly what they are. It's important to understand these aren't so much as, they're not alternatives to binary fission. They're processes that occur in bacteria that allow genetic recombination despite binary fission. So we have binary fission occurring. Binary fission is producing these progeny that are essentially genetically identical except for chance mutations. However, these 3 processes, transformation, transduction, and conjugation, are inducing genetic recombination in these cells that would otherwise be fundamentally genetically identical. Firstly, we have transduction, and I have an image here of a phage. You may know what a phage is. If you don't, I'll give a quick explanation just as an aside, because we need to know to understand transduction. A phage is short for bacteriophage, and a bacteriophage is a type of virus that specifically infects bacteria. So it specifically infects bacterial cells. The way it infects is it lands on the surface of the bacterial cell using what these protrusions that look like legs, and then it injects its genome. The genome is stored in its head at the top. Again, it's not really a head, it's just a protein capsid. Then the genome again is injected into the bacterial cell. Once it's inside the bacterial cell, it's replicated and expressed, not dissimilar to how other viruses infect host cells. It is important to remember that bacteriophage is a virus. It's just a type of virus that has evolved to specifically infect bacteria. So what do these bacteriophages have to do with recombination in bacteria? What role do they have in the genetic recombination of bacteria? The answer is that when these bacteriophages inject their genome into bacterial cells, they can change the fundamental genetic composition of the cell they're infecting. It's almost the point, right? They're introducing new genetic material into the bacterial cell. a lot of that genetic material will just be the bacteriophage's own genome. So it will be genes that, when expressed, will allow for the production of new bacteriophages. So progeny, so it's just part of the bacteriophage's replication cycle. However, some of the genes can have other effects. So for example, some of these genes can induce the bacteria to become pathogenic. There are a large number of bacteria that, under normal conditions, if they're present in the human system, they don't really cause disease. However, if they are transduced by a bacteriophage that carries a specific gene, they can become pathogenic, because by having this gene, they gain the ability to cause disease. The clearest example is Streptococcus pyogenes. So Streptococcus pyogenes is the causative agent of scarlet fever. However, only strains of Streptococcus pyogenes that have been transduced with a particular gene are able to cause scarlet fever. So that's why this matters. It can seem a bit weird, it can seem an unusual way by which the genetic material of a bacteria is altered, and it is strange. However, it definitely happens. It's really important. Scarlet fever is evidence for that, and it's not the only example. When we look at the infectious disease lectures, we'll actually look at quite a few examples of different bacteria that are capable of inducing disease in humans only when they are transduced by a bacteriophage. Okay, what we have here is transformation. Transformation is another method of genetic recombination, so it's another way by which bacterial cells can gain new genetic information. Very different to transduction. First, you'll notice there's no bacteriophage present in the diagram. That's because bacteriophages aren't involved at all. The role of bacteriophages in bacterial genetic recombination is essentially confined to transduction. Transformation essentially just means the ability of bacteria to uptake DNA fragments in their environment. In biological systems, there's often a fair bit of DNA just randomly floating about from when cells have died. Often cells die and they lyse, and their genetic material just gets released into the environment. When we have bacterial populations where we have a lot of bacteria living and growing in close proximity, this happens even more often. So it's perhaps not surprising that bacteria have the ability to uptake genetic material that is in their immediate environment. It actually makes sense as a reasonably good way to gain new genetic material. Once this genetic material is absorbed by the cell, it's then often integrated into the genome of the bacteria. It can't really integrate randomly. It has to integrate at a site where there's complementary bases, so there's actually base pairing going on. However, it does happen. It's a fairly well-established process. Once this has happened, it means that the genetic material can be retained because it's part of the bacterial genome. If you compare that to just a bit of almost random DNA just floating around the cell, not really being transcribed or translated, that's not going to be retained over generations. However, once the genetic information is integrated into the actual genome in the nucleoid, then it's retained over generation and generation and generation. Okay, conjugation is the final method of genetic recombination we want to consider. So we talked about pili earlier, and I mentioned sex pili. Sex pili are a specific type of pili that are involved in conjugation. I also refer to them as conjugative pili. We tend to use sex pili when we're talking specifically about E. coli. When we're talking about pili that do this process in general, We tend to use the term conjugative pili instead. The principle is exactly the same. It's just we have those specific terms for E. coli. So pili is, as we talked about, a protrusion from the cell. It's composed of pilins. In this context, what it's doing is connecting with another cell and allowing the transfer of genetic material into this other cell. Really cool, really interesting. We're getting the physical transfer of genetic material through the pili into a second cell. There's something called the F factor. The F factor is important as well because the only way this process can occur is if the F factor is present in the donor cell. So we have cell A producing a conjugative pili to cell B and transmitting genetic material through the pili into, or through the pilus, if we're talking about a single pili, through the pilus into the recipient cell. This can only happen if the donor cell has the F factor. The F factor is on a plasmid. If you remember, we talked about those plasmids. Sorry, not always on a plasmid. It's usually on a plasmid. In normal conditions, there are exceptions. And this F factor basically allows this process to occur. So we need to remember that donor cell has the F factor, allows it to produce pili or a pilus, which connects with the recipient cell, and then genetic material can be transferred through the pili into the recipient cell. And we can see that process demonstrated diagrammatically here. We see there's two cells linked by a mating bridge. This mating bridge is the conjugative pili. You see we have a donor cell and a recipient cell, and the donor cell has the F plasmid, also called the fertility plasmid, also called the fertility factor, and that's what defines it as the donor cell. As we progress across this diagram, you'll see a copy of the fertility plasmid is being fed through the mating bridge, so through the conjugative pili into the recipient cell. The recipient cell at the start is F minus. It doesn't have the F factor, it doesn't have the F plasmid. That's why it's able to act as a recipient cell. However, by the end, when it's received a full copy of the fertility factor, of the fertility plasmid from the donor cell, it is then defined as F plus because it has a fertility factor itself. There's just a little bit more to the story of conjugation. So we've covered most of it. It's just this little add-on we need to discuss. So there's cases where there are HFR cells. HFR stands for high frequency of recombination. The situation with these cells is slightly different to the situation with the F plus cells because these cells are slightly different themselves. In HFR cells, the fertility factor is not contained on a plasmid. It's actually integrated into the genome of the cell. So the main genome within the nucleoid contains the fertility factor. What this means is when this cell forms a mating bridge with an F minus cell, so when a HFR cell forms a mating bridge with an F minus cell, it then attempts to transfer its whole genome into this F minus cell. That's because that's just what the F factor does. That's what the fertility factor does. That's what it encodes. It encodes the capacity to transfer this genetic material. However, because it's part of the cell's main genome, it drags along the rest of the genome with it. This is far too much genetic information to be transferred across a pili usually. So what happens is some of it gets transferred across, potentially a lot of it gets transferred across, but then the connection between the two cells breaks down. It's just not a sustainable transfer. It's just a huge amount of genetic information. So eventually the connection via the mating bridge breaks. In this situation, what we usually have is a portion of the HFR genome, including the fertility factor, floating around the recombinant bacteria, so floating around the interior of the F- cell. So again, this is the F- cell, which the fertility factor and a portion of the donor cell's genome has been transferred into. It's not a plasmid, so it can't sustain itself in this cell. It can't replicate. It can't be expressed. It can't be transcribed or translated. Ultimately, it's usually broken down by cellular enzymes, because that's what happens to random bits of DNA that are floating around a cell if they're not circularized, so if they're not plasmids. However, while this is happening, or before this is happening, you can get bits of this donated genome recombining into the main genome of the donor cell. So, sorry, not the donor cell, the recipient cell. So we're in the recipient cell now, and we have a chunk of genetic information that's been transferred across from the donor cell. Most of this genetic information is going to be broken down by enzymes. However, little bits can recombine and therefore be retained in the main genome of this cell, the recipient cell. Okay, so that's bacterial reproduction covered, hopefully. If anything wasn't clear, again, just head back to the resources we have in the module space. Look at the course textbooks as well. There's nothing super complicated there. Some of it just maybe takes a little bit of time to wrap your head around, but hopefully it will all make sense eventually with a little bit of study. Finally, I want to talk about bacterial metabolics. Again, there's some technical concepts here, but nothing too difficult to understand, nothing that I think anyone will actually really struggle to understand. If any of it isn't clicking, just do the study you need to do. So just spend time trying to understand it, spend time reading around it until everything kind of fits into place. Here we have metabolics. These are kind of an outline of the key areas we're thinking about when we're talking about metabolics, and we can really split the area into two. We think of carbon source and energy source. So all bacteria need a carbon source. This is what they need to create organic molecules. They need carbon, carbon-based life. They need to get this carbon from somewhere. They also need an energy source. So to complete the processes that define life, bacteria, it takes energy, so they need energy. So where are they going to get this energy from? And what we see is carbon and energy each come from two potential sources, and different bacteria will use either one of these sources. So for carbon sources, one source is CO2. So some bacteria will obtain carbon themselves from CO2, so they'll just pull it from CO2. Heterotrophs will do the opposite. They will feed on external carbon sources, so they will feed on organic molecules which already have kind of complex structures based around carbon, and that's where they're getting their carbon from rather than raw CO2. Autotrophs are the term we use to the groups that obtain carbon from CO2. Heterotrophs are a term we use to describe the groups that obtain their carbon from other sources, so external carbon sources. Auto means self. Again, autotroph, they're kind of they're not relying upon other organisms. They're getting the carbon themselves straight from CO2. Heterotrophs, they're feeding on carbon molecules, they're carbon containing compounds that are being produced by other organisms, and they're hetero meaning other, and hence the term. Next up, we want to think about energy source. And again, as I said, there's two ways that bacteria can obtain energy, two sources they can derive energy from. First, we have the phototrophs. These derive energy from light. Plants, obviously not bacteria, but they are a well-known organism that derives their energy, or a group of organisms that derive their energy from light. Other bacteria are chemotrophs, so these will obtain their energy through the respiration of chemical compounds, so they're relying upon chemical energy, and they'll obtain these chemical compounds and they'll obtain energy from these chemical compounds. By considering whether or not a bacteria is an autotroph or a heterotroph, and a phototroph or a chemotroph, we can classify microorganisms. As I said, all bacteria are either autotrophs or heterotrophs, and all bacteria are either phototrophs or chemotrophs. So we can compose a nice little flow diagram onto which all bacteria should fit. I don't like this flow diagram. Some students do, so that's why I've included it. This flow diagram first looks at energy source, then it looks at carbon source. That doesn't make sense in my head. In my head, it makes sense to look at carbon source, then look at energy source. I don't even know why. I think maybe it's just the way I learned it a long time ago. But I'll talk through this diagram and then we'll look at it flipped. Whichever makes the most sense, just rely upon that. As I said, the information is the same. It's just how it's being presented. Okay, so in this diagram, first off, we're thinking energy source. So we'll take a particular bacteria and we'll ask, does it obtain its energy from chemical compounds, or does it obtain its energy from light? If it obtains its energy from light, it's a phototroph, and we go down the right arm. If it contains its if it obtains its energy from chemical compounds, then we go down the left because it's a chemotroph. And we'll stick with the left for this example. So we're working with a chemotroph. Then we consider its carbon source. So does it obtain its carbon from organic compounds or directly from CO2? If it obtains its energy from organic compounds, then it is a heterotroph. If it obtains its carbon from directly from CO2, then it's an autotroph. However, we already know that it's a chemotroph referring to the energy source, which is chemical. So therefore, we combine the terms and we say it's either a chemoheterotroph or a chemoautotroph. So the chemo part of the word is established because we know it's a chemotroph, and then we're just adding in the hetero or the auto, dependent upon whether it obtains its carbon from organic compounds or from CO2. Now we'll go and explore the other arm of the diagram, and we'll say we're looking at a phototroph. So this is a bacteria that obtains its energy from light, phototroph. This phototroph can either obtain its carbon from organic compounds or directly from CO2. So again, this is just a mirror of the other side of the chart. If it obtains its carbon from organic compounds, it's a heterotroph. If it obtains it from directly from CO2, then it's an autotroph. Again, we already know it's a phototroph because we're on this side of the diagram. It obtains its energy from light. So if it obtains its carbon from organic compounds, it's a photoheterotroph. If it obtains its carbon from CO2 directly, then it's a photoautotroph. Again, auto means itself. It's obtaining its carbon from CO2 directly itself. Hetero means something else, which means it's relying upon organic compounds, complex organic compounds that other organisms have already made, that have already fit together. So it seems like a lot of big words and a couple of difficult concepts, but really it completely makes sense once you, again, just spend a little bit of time thinking about it. And here we have the diagram that I was referring to when I said I like a slightly different diagram, which is it's essentially the last diagram just flipped on its head a little bit. So we're looking at carbon source first, then looking at energy from light. The information is exactly the same, and if you're looking at any particular bacteria and you're asking these questions on this graph, you'll end up at exactly the same definition, same classification as you would on the last graph, just preference. So the first question is carbon obtained from elsewhere, so from other organic compounds, or is it directly obtained from CO2? So specifically the question is carbon obtained from elsewhere? If it is, we're dealing with a heterotroph because it's obtaining its carbon from other organic compounds. It's not directly obtaining it itself from CO2. So we'll go down the right arm. Yes, carbon is obtained from elsewhere. It's a heterotroph. Is energy obtained from light? Yes, energy is obtained from light. Then it's a photoheterotroph. Is energy obtained from light? No. Then it's a chemoheterotroph, because if it's not obtaining its energy from light, it's obtaining it from chemical compounds. Okay, back to the start. So again, we can ask, is carbon obtained from elsewhere? In our example now, we'll think about a microorganism that doesn't obtain its carbon from elsewhere. It's an autotroph. It obtains carbon directly from CO2. So the answer is no. Okay, then we can ask, does it obtain its energy from light? If the answer is yes, then it's a photoautotroph. If the answer is no, then it's a chemoautotroph because it must obtain its energy from chemical compounds. So I hope that made sense. Again, if it doesn't, textbooks will be really useful. Okay, here we have a little bit more information on chemoheterotrophs, mainly because these are the most it's the most common category. Most bacteria can be classed as chemoheterotrophs. So because chemoheterotrophs are the most common category, it's worth talking about them a little bit and worth making sure we understand what's going on with them. Hopefully we've defined them. The real main extra bit of information I want to give is that for most maybe not most, maybe most, for a large number of chemoheterotrophs, the chemical compounds from which they derive their carbon are the same chemical compounds as those from which they derive their energy. So we know chemoheterotrophs derive their carbon from organic molecules, usually sugar, glucose. Same for light. We know they obtain their light. Same for energy. We know they obtain their energy not from light, but from chemical compounds. Again, sugar, glucose. Most chemoheterotrophs are obtaining their carbon and their light from the same source, and that source is a chemical compound, and that chemical compound is often glucose, often a sugar, specifically glucose. Okay, so we can even split these chemoheterotrophs up into two groups as well, because we can ask, okay, where do they get their glucose from? The parasites, these are obtaining their glucose from other living organisms, so they're feeding on other living organisms. The saprophytes, however, are obtaining their sugar from dying organisms, or dead organisms that are decaying. So saprophytes play a big role in the decay of dead organisms because they're feeding on them, essentially. Okay, a very good example is E. coli. Again, the carbon source and the energy source isn't always glucose. It's just we tend to think of glucose as the most obvious one, and there's a lot of bacteria that do use glucose. E. coli is one example. So it's a heterotroph, a chemoheterotroph specifically. So it's obtaining its carbon and its energy from chemical compounds, and the chemical compounds it uses for both of these processes are glucose. So it obtains its carbon from glucose and it obtains its energy from glucose. Just a quick look at some of the other groups, just so we have a few examples in our head. Chemoautotrophs. So again, chemoautotrophs are gaining their energy from chemical compounds. However, they're gaining their carbon from a different source. They're gaining their carbon directly from CO2. And we see this with the Pseudothiobacillus thiooxidans. So this is a bacteria that, again, utilizes CO2 for its carbon. However, it doesn't use light for energy. It obtains its energy from a chemical source, specifically from the oxidation of inorganic sulfur. So it's just a chemical process. Thiooxidans is quite interesting, and we'll talk about it in lectures towards the end of the module, I imagine, when we're discussing the usefulness of bacteria. It's found in copper mines and it's found in sewage systems, and the reason is because it thrives in these sulfur-rich environments, which isn't surprising given how it derives its energy. The reason why we think of it as being relevant to the last portion of the module is because then we're talking about using microorganisms as tools, and this is an example of a microorganism that can be used in that way because it can be potentially used in bioleaching. And bioleaching essentially means using biological organisms to leach, so to absorb various chemical compounds from the environment. That's one of the potential uses of thiooxidans. We're going to go into it in a lot more detail towards the end of the module. Okay, an example of a photoautotroph would be the photosynthetic bacteria. So these are bacteria that, again, they're deriving their carbon from CO2. However, unlike the chemoautotrophs, they are deriving their energy from light. They photosynthesize, but they don't photosynthesize in exactly the same way as plants or plant cells, because they don't have chloroplasts. They have thylakoid compartments, and this is where the photosynthesis takes place. So it's the overarching purpose of the process, and in some ways the process overall is the same. It's still photosynthesis. The same thing is still happening. It's just the way it's done is slightly different, and this kind of reflects the fact that we're talking about different cell types that belong to different kingdoms of life. So it's understandable that the details of how the process is done, where in the cell it's done, would differ. And here's just an example. We can see the thylakoid membrane here, and the thylakoid membrane is specifically where this process is happening. If you're comparing this cell here to a bacterial cell, you'll see it's just completely different cell types. So again, don't confuse the two cell types just because they both undertake photosynthesis. Details are very different. Finally, we have the photoheterotrophs. These, there's nothing particularly interesting about them apart from the fact that they are photoheterotrophs. The principal examples would be the Rhodospirillum and the Rhodobacter. These are mostly just known because they're photoheterotrophs, though that's not absolutely true. But our principal interest in them is the fact that they're photoheterotrophs. So we know the term photoheterotroph by now. They're heterotrophs, so they're obtaining their carbon from organic compounds, and the photo in their name means that they are obtaining their energy from light. The way in which they obtain their energy from light is slightly different to the photoautotrophs because the photoheterotrophs conduct anoxygenic photosynthesis. So that means they're conducting a form of photosynthesis that doesn't result in the production of oxygen. And that's all we really need to know about them. Finally, we want to touch on oxygen requirements. We're talking about the bacteria metabolics. We need to understand that different bacteria have different requirements for oxygen. So different bacteria can only grow in certain levels of oxygen. This is a summary of what the different levels, so what the different oxygen requirements of bacteria are defined as. Again, we mentioned it the other day. This is just to qualify the statements. We have aerobes and we have anaerobes. Aerobes, these grow with oxygen, they don't grow without oxygen, so they need oxygen to grow. Anaerobes are the opposite. They grow in the absence of oxygen, but they don't grow in the presence of oxygen. Facultative, these have the capability to grow in both, so they can kind of switch, and you'll see the term facultative aerobe and facultative anaerobe. This is just adding some degree of preference, so it's saying they have a preference for aerobic conditions or anaerobic conditions. Indifferent refers to bacteria that are essentially indifferent to the presence of oxygen, as the name suggests. So they can grow in the presence or absence of oxygen. It doesn't really make much of a difference to them. It doesn't affect their metabolics. Microaerophilic, we tend to use this term to refer to bacteria that grow in, they can grow in the presence of oxygen. They prefer low levels of oxygen, lower than what we see in the atmosphere, certainly. So in normal oxygen conditions, you do see some growth. However, it's not their preferred growth environment. Finally, just some examples here. Again, I don't think I need to say anything about them. I've picked examples that you should know, or you should know by the end of the module. So these are bacteria that we'll talk about at some point in the module. Many of these during the infectious disease lectures. Okay, so I hope that was useful. Again, if you've had any significant issues understanding anything, the first thing you should do is go back and listen again. If that doesn't help, course textbooks, other resources. If that doesn't help, just feel free to shoot me a message. I'm always happy to answer an email or a Teams message. Otherwise, I'll see you at the next lecture. Thanks very much.